Hi, I'm George. I'm Scientifica's Scientific Content and Marketing Executive, and I'm here with David Wyllie, who is a Professor at the University of Edinburgh and the Director for the Centre for Integrative Physiology. Hi.
Hi.
Could you please tell us a little bit about your research and what you're hoping to learn?
So at Edinburgh we run a very large collaborative programme of research which is focused in three main areas. These are in preclinical models of neurodevelopmental disorders. In the study, which has been a long-standing interest of mine, the study of structure function studies of ligand-gated ion channels, most notably N-methyl-D-aspartate or NMDA receptors. And more recently, I guess in the last 5 years, studies which involve the use of human pluripotent stem cells to model both neurodevelopmental and neurodegenerative diseases, where we generate from these pluripotent stem cells both neurons and glia. So overarching in this is work which looks to see, what is the dysfunction that is caused either in our model of a neurodevelopmental disorder, or what perhaps is seen at dysfunction in terms of a cell, pluripotent stem cell derived neuron and glia, to see what changes in cellular function occur. And the key question that we're interested in is, how do these manifest, these changes in either cellular function, manifest at levels which will impact function and ultimately, obviously, in the brain, so at the circuit level, and ultimately at behaviour. So in order to do that, we need to take this multidisciplinary approach. I'm obviously heavily involved in the electrophysiological studies, but combined with that, we've got imaging studies as well, biochemical assessments. So all of this sort of pulls together in this sort of large collaborative program to advance in terms of our understanding in these areas, whether it be single channel biophysics, models of, for example, Fragile X syndrome or haploid insufficiency in SYNGAP1, or using pluripotent stem cells to model ALS.
And what laboratory techniques do you use to carry out your research?
So the main laboratory technique that I've been involved with and has been throughout my career is in electrophysiology, and these techniques involve conventional electrophysiological techniques such as whole cell recording from brain slices, where you use voltage clamp, current clamp recordings. We may want to look at the activity of individual ion channels, so we use single channel patch clamp recordings from isolated membrane patches. We also combine our whole cell electrophysiology with imaging in studies, so we use multiphoton imaging combined with electrophysiology, and then in collaboration with others, in vivo recordings and combined imaging as well. So the heavy focus for us is always on the electrophysiology. We also carry out using other techniques, and occasionally some sharp intracellular recordings or some field recordings as well to assess aspects of synaptic plasticity.
Great, thank you very much.